Okay. So yeah, I'm going to talk to you guys about the contents of my master's thesis, which was titled Mapping Proteomic Composition at Excitatory Synapses in the Cerebellar Cortex. So these are two papers, one recently published and the other recently accepted from our lab. The first one is Matthias's paper, which I'm sure you guys will hear about in his presentation. But that one provides a little bit more details on the protocol that I used, the adapted version of MAP. And the second one is my paper, which contains the results that I'm going to talk about today. That one is accepted but not yet published. My thesis is online under the same title. If you guys are curious about that before it's published, there's a version of the paper in my thesis. So yeah, just as an overview, sorry if I'm talking too loud into this mic. I feel like I'm anyways, as an overview of what I'm going to talk about today, my study used an adapted version of MAP to identify differences in neurotransmitter receptor, scaffolding protein, and enzyme composition at different synapse types within the cerebellar cortex. And I'm going to present my results to you in two different parts. The first part, we'll be looking at the composition of excitatory postsynaptic proteins at parallel fiber, climbing fiber, and molecular layer interneuron synapses in the molecular layer. And the second part, we'll be looking at the composition of excitatory postsynaptic proteins at synaptic versus extrasynaptic sites in granule cell glomeruli. So I'm just going to start out by giving you an overview of the cerebellar cortex and the circuitry there. So the cerebellar cortex is made up of three different layers. The innermost layer is the granule cell layer. Let's see if I can get a laser on here. Right here. There's then a middle layer, which is a single cell layer of Purkinje cell bodies, which is the Purkinje cell layer. And then the outermost layer is the molecular layer. The cerebellar cortex receives two different types of inputs, which are mossy fibers and climbing fibers. The climbing fibers originate in the inferior olivary nucleus and project up and wrap around the dendrites of the Purkinje cells. Inputs from a single climbing fiber are enough for a Purkinje cell to reach action potential. The other type of inputs coming into the cerebellar cortex are mossy fibers, which originate from multiple different brainstem nuclei. And the mossy fibers come into the granule cell layer and create excitatory synapses with granule cells at these specialized structures called glomeruli. Granule cells then project parallel fibers up into the molecular layer where they bifurcate and travel perpendicular to the Purkinje cell dendritic arbor. Inputs from multiple parallel fibers are needed for a Purkinje cell to reach action potential. And these parallel fibers also create excitatory synapses with molecular layer interneurons. These molecular layer interneurons have originally or classically been divided into two different types. And those are basket cells, that are located in the inner 2/3 of the molecular layer or in the proximal molecular layer. And they tend to create synapses, inhibitory synapses onto the cell bodies of Purkinje cells. And there are also stellate cells, which are located in the more distal 2/3 of the molecular layer. The major excitatory synapses in this circuit that we'll be focused on throughout this presentation will include mossy fiber to granule cell, parallel fiber to Purkinje cell, parallel fiber to molecular layer interneuron, and climbing fiber to Purkinje cell. So these excitatory synapses in this circuit are all made up of the same basic components. These components include a presynaptic terminal, where glutamate is packaged into vesicles via a vesicular glutamate transporter before fusing with the cell membrane in the active zone via calcium-dependent release with the support of the snare complex, as well as multiple different scaffolding proteins, which include ELKs and RIM, which are located at all presynaptic sites. Once glutamate is released into the synaptic cleft, it binds with receptors on the postsynaptic cell, and the postsynaptic site also contains the postsynaptic density, where many signaling and scaffolding proteins that are important in the function of the synapse are located. These include, but are not limited to, all of the proteins that you can see on this diagram here. This is an extremely simplified version of the excitatory synapse. In reality, there are thousands of these postsynaptic density proteins that are all important in the function of the synapse, but these are the ones that I have chosen to focus on. So in this little complex here, we have PSD95 that binds with receptors. In the case of AMPA receptors via TARPs, SYNGAP and SAPAP then bind with PSD95. SAPAP links PSD95 to SHANK, which then connects the postsynaptic density to actin via cortactin. Each of these synaptic components has a different role at the synapse, and many are protein families in which each member has a slightly different role. And the excitatory synapse has been historically very difficult to study due to its small and dense and complex nature. Previous studies looking at the composition of the excitatory synapse have mainly used electron microscopy, which is an incredibly laborious technique and involves identification of synapses via structure. So this is where an adapted version of magnified analysis of the proteome comes in. As I mentioned earlier, the excitatory synapse, particularly the postsynaptic density, is incredibly small and challenging to study. The small and dense nature of the structure provides multiple difficulties in its visualization. These include resolution as well as antibody accessibility. I used an adapted version of magnified analysis of the proteome to overcome these problems. MAP involves cross-linking the proteins in the tissue with an acrylamide gel matrix before clearing and expansion, which clears the lipids from the tissue, making it transparent, as well as expanding the tissue by approximately 4 times. This both provides greater resolution with a confocal microscope, as well as greater antibody access. This allows me to acquire single synapse resolution with an increased number of available antibodies. So these images here are just showing exactly how much resolution I'm gaining when I use MAP. So this first image here is a confocal image stained with synapsin 1 in green and PSD95 in red. And this here is the cerebellar granule cell layer. So all of these little clumps here are the glomeruli. And what you can see when I zoom in here, each glomerulus is just kind of a blur of red and green. And when we look at images that I've taken with MAP processed tissue, this is one of those glomeruli. So each of these clusters is this whole area here. And you can see when I zoom in even further, I have in this image PSD95 in green and ELKs in red. So you can see I can actually see the presynaptic and postsynaptic sites separately in these images. So yeah, I'm gaining a huge amount of resolution and the antibodies look quite nice. So moving along to part 1 of my results. This one looked at composition of excitatory postsynaptic proteins at parallel fiber, climbing fiber, and molecular interneuron synapses in the molecular layer. And just as a summary of the molecular layer and to show the markers that I used, to mark parallel fiber to Purkinje cell synapses, I used GLUT2, which is a member of the GLUT2 family of ionotropic glutamate receptors and is known to be expressed only at parallel fiber to Purkinje cell synapses. For climbing fiber to Purkinje cell synapses, I used vGlut2, which is a vesicular glutamate transporter, which is only expressed at the presynaptic terminals of climbing fiber to Purkinje cell synapses. And PSD95 is located at the postsynaptic site of the climbing fiber to Purkinje cell, and molecular and parallel fiber to molecular layer interneuron synapses. However, it's also located at a very small subset of parallel fiber to Purkinje cell synapses. So just showing each of these, the staining of each of these markers, I have here shown GLUT2, ELX, and PSD95, and we can see in the merged image that GLUT2 and PSD95 tend to localize to different synapses, both of which are ELX positive. So I used GLUT2 positive synapses to define parallel fiber to Purkinje cell synapses and GLUT2 negative PSD95 positive synapses to define non-parallel fiber to Purkinje cell synapses. And this staining with a transgenic mouse line, which shows interneurons, mainly in the cortex, but does show a subset of interneurons in the cerebellum, shows that PSD95 is located along the interneuron dendrite, and GLUT2 is mainly localized away from that dendrite. And finally, I have staining of GLUT2, PSD95, and VGLUT2. And we can see in the merged image that with those big presynaptic VGLUT2 terminals, we can see that the postsynaptic sites are PSD95 positive, and GLUT2 is not located at those sites. It's located at the parallel fiber to Purkinje cell synapses. And so for this portion, I used VGLUT2 positive, PSD95 positive to define climbing fiber to Purkinje cell synapses, and VGLUT2 negative, PSD95 positive synapses to define non-climbing fiber to Purkinje cell synapses. Okay, so onto some results from the parallel fiber to Purkinje cell versus non-parallel fiber to Purkinje cell synapses. So this was co-staining with GLUT2 and PSD95. And we can see in the first two panels with pan-AMPA and GLUA2 that AMPA receptors are mainly localized to synapses that are PSD95 positive. So those would be the parallel fiber to molecular layer interneuron, as well as climbing fiber to Purkinje cell synapses. And they are not really localized with GLUT2. So there is less AMPA receptors at those parallel fiber to Purkinje cell synapses. And in the bottom two panels, you can see SHANK1 and SHANK2, and we can see the opposite pattern with SHANK1 and SHANK2, where GLUT2 positive synapses are also positive for SHANK1 and SHANK2, where PSD95 positive synapses are not as often positive for SHANK1 and SHANK2. So I used Arevis to quantify the images that I showed in the last slide. And I was able to get density and intensity measures from Arevis. And so the first panel here shows the fraction of parallel fiber to Purkinje cell synapses and non-parallel fiber to Purkinje cell synapses that were positive for each antigen of interest. A three-way ANOVA showed significant differences between antigens and between synapse types, but not between the proximal and distal regions of the molecular layer. And we can see in these images that, as in the previous images, pan-AMPA and GLUA2 were localized more to the non-parallel fiber to Purkinje cell synapses than to the parallel fiber to Purkinje cell synapses. And we can also see that SHANK1 and SHANK2 were localized far more at parallel fiber to Purkinje cell synapses, where SHANK3 showed a similar distribution across both synapse types. And the second panel here shows the fraction of parallel fiber to Purkinje cell to non-parallel fiber to Purkinje cell integrated intensity. And in this graph, a two-way ANOVA showed significant differences between antigens, but not between the proximal versus distal regions of the molecular layer. And post-hoc testing showed that SHANK1 and SHANK2 had a significantly higher ratio compared to the other antigens, indicating that they were both brighter at parallel fiber to Purkinje cell synapses than non-parallel fiber to Purkinje cell synapses. So as I mentioned earlier, there's a small subset of these parallel fiber to Purkinje cell synapses that are positive for PSD95. So I decided to take a closer look at which of these, at the parallel fiber to Purkinje cell synapses that were positive for PSD95. So here I show similar measures to the last slide, with A being a fraction of antigen positive, parallel fiber, or PSD95 positive and PSD95 negative parallel fiber to Purkinje cell synapses. And I actually found here that all of the antigens had a greater density at the PSD95 positive parallel fiber to Purkinje cell synapses than at the PSD95 negative parallel fiber to Purkinje cell synapses. A three-way ANOVA showed significant differences between antigens, as well as parallel fiber to PSD95 negative to PSD95 positive parallel fiber to Purkinje cell synapses, but did not show significant differences between the proximal and distal regions. And in the second graph here as well, I showed the ratio of PSD95 positive to PSD95 negative integrated intensity. And all of these were greater than 1, indicating that they were all brighter at PSD95 positive parallel fiber to Purkinje cell synapses. But post-hoc testing showed that PAN-AMPA, GLU-A2, and SAPAP1 had all had greater ratios than all of the other antigens. So this indicates that PSD95 positive parallel fiber to Purkinje cell synapses may be larger or more dense than PSD95 negative parallel fiber to Purkinje cell synapses. So just to bring us back to the markers that I used, I'm now going to move on to talking about climbing fiber to Purkinje cell versus non-climbing fiber to Purkinje cell synapses, which I used vGlut2 as well as PSD95 to mark. And so we can also see differential distribution of synaptic proteins at climbing fiber versus non-climbing fiber synapses. In the first panel, we can see that SHANK1 is localized more to the non-climbing fiber synapses and at very low levels at climbing fiber synapses. SHANK1 is not really seen at very many climbing fiber synapses. And we can see the opposite pattern with SHANK2, where SHANK2 is highly expressed at those climbing fiber synapses and expressed at less of the non-climbing fiber synapses. SYNGAP shows a very low expression, but similar to SHANK1 is not really localized to those climbing fiber to Purkinje cell synapses. So these results show the fraction of climbing fiber versus non-climbing fiber synapses positive for each antigen. The two-way ANOVA showed significant differences between antigens, but not between climbing fiber versus non-climbing fiber synapses. We can see that AMPA receptors, SHANK3 and SAPAP1 are highly expressed at both synapse types, where SHANK1 and SYNGAP are lower at climbing fiber synapses, and SHANK2 is lower at non-climbing fiber synapses. In part B, there's a ratio of climbing fiber to non-climbing fiber integrated intensity, which showed significant differences between antigens via a one-way ANOVA, again with SHANK1 having a particularly high ratio, and SHANK1 and SYNGAP with particularly low ratios. So this is just a summary of my overall findings from the molecular layer. These results confirmed a distribution of AMPA receptors to parallel fiber to molecular layer interneuron and climbing fiber synapses, which has been previously shown via electron microscopy and showed a similar distribution of SAPAP1. The 3 SHANK proteins were differentially expressed, with SHANK1 at parallel fiber to Purkinje cell synapses, SHANK2 at parallel fiber to Purkinje cell and climbing fiber to Purkinje cell synapses, and SHANK3 at all 3 synapse types. These results showed that there are not only cell type specific differences in expression, but also input specific differences between parallel fiber to Purkinje cell and climbing fiber to Purkinje cell synapses. The mechanism of these input specific differences is not known. However, GLUD2 has been shown to have a regulatory role on AMPA receptors. With GLUD2, with GLUT2 knockout resulting in increased AMPA receptors at these synapses, at parallel fiber to Purkinje cell synapses, sorry. It is not known whether other synaptic components are regulated in a similar way. In addition to this, I also found no significant differences between parallel fiber to Purkinje cell versus non-parallel fiber to Purkinje cell synapses in the proximal versus distal regions of the molecular layer. Given the morphological classification of molecular layer interneurons into basket and stellate cells, differences in expression at non-parallel fiber to Purkinje cell synapses would have been expected. These results may be more consistent with a more recent classification of molecular layer interneurons, which includes two molecularly and electrophysiologically distinct groups spread across the molecular layer. So moving on to the second part of my results, the composition of excitatory postsynaptic components at synaptic versus extrasynaptic sites in granule cell layer glomeruli. So these glomeruli are a very unique structure in the brain. We can see the purple coming through the center is a mossy fiber terminal, which creates multiple synapses with granule cell dendritic digits. There have also been, in my beginning studies, at the beginning of my research, I started finding these PSC95 positive clusters that were negative for ELKs. And at first we had no idea what was going on because the PSC95 antibody we have is quite good. And we were thinking maybe it was a different isoform of ELKs that our antibody didn't pick up. We then found reports of PSC95 positive adherens junctions or like extrasynaptic sites within these cerebellar glomeruli. There has been controversial evidence of about the localization of NMDA receptors, with some studies reporting that NMDA receptors are mostly extrasynaptic in the glomeruli, and other studies reporting that NMDA receptors are purely synaptic in the granule cell layer glomeruli. So I looked into the localization of receptors, scaffolding, and signaling proteins at synaptic versus extrasynaptic sites using ELKs and PSC95 as markers. So I used ELKs positive, PSC95 positive clusters to mark synapses, and PSC95 positive, ELKs negative clusters to mark extrasynaptic sites. And so we can see with these two images here, I've stained with ELKs and RIM, which should be localized to all presynaptic sites. So we should see all the synapses here. And we can see that GLUN1 is localized mainly away from those ELKs positive, RIM positive clusters. So yeah, we can see some synapses over here. And these GLUN1 positive clusters don't contain ELKs or RIM, indicating that they may be extrasynaptic. And in the bottom panel here, I have PSC95 stained with ELKs and RIM. And we can see that PSC95 is located with ELKs and RIM, but it's also located at other sites away from ELKs and RIM. So here I have staining of PAN-AMPA as well as GLUN1, co-stained with ELKs and PSC95. And we can see in the image of PAN-AMPA on the top, we can see that PAN-AMPA is mainly localized to ELKs positive sites and is mainly at synapses in the cerebellar glomeruli. Where if we look at the bottom panel, we can see that GLUN1 is mainly localized away from ELKs positive synapses and at PSC95 positive extrasynaptic clusters. So I again quantified these images using Arevis, and these results show the fraction of synaptic, this first graph here, shows the fraction of synaptic versus extrasynaptic sites that are positive for each antigen of interest. A two-way ANOVA showed significant differences between antigens as well as between synaptic versus extrasynaptic sites. And post-hoc tests showed that PAN-AMPA and GLUA2 were present at significantly more synaptic sites, where GLUN1 was present at significantly more extrasynaptic sites. The following graphs show ratios of synaptic to extrasynaptic integrated intensity per cluster and per field. The per cluster measures did show significant differences via a one-way ANOVA. However, the only ratio, did not. The only antigen with a ratio below 1 was GLUN1. Measures per field showed significant differences between antigens via one-way ANOVA, and post-hoc tests showed significantly lower intensity of GLUN1 and SYNGAP at synaptic sites, as well as significantly higher intensity of pan-EMPA at synaptic sites. So there was also reports of these adherens junctions being M-cadherin positive. So I then did further staining to look at whether these M-cadherin positive adherens junctions were positive for the antigens that I was looking at. And these images show, as we can see here, again, pan-AMPA in the top panel was not localized with extrasynaptic PSD95 or with M-cadherin. It was localized to the PSD95 positive synapses. Where GLUN1 and SYNGAP both showed colocalization with M-cadherin as well as PSD95 at these extrasynaptic sites, indicating that GLUN1 and SYNGAP are both localized together at extrasynaptic sites in the cerebellar glomeruli. So this shows a summary of my granule cell layer results. These granule cell layer results showed differential expression of AMPA and NMDA receptors at synaptic versus extrasynaptic sites, with AMPA receptors at synaptic sites and NMDA receptors at extrasynaptic sites. SYNGAP showed a similar expression pattern to NMDA receptors, with both colocalizing with M-cadherin at adherens junctions. Though previous studies have provided contradicting results on the localization of NMDA receptors, this study shows that they are mainly localized to extrasynaptic sites, which is supported by electrophysiology data, which shows activation of NMDA receptors via spillover, indicating a functional significance of extrasynaptic clusters. NMDA receptors have also been shown to have a role in cell-to-cell adhesion via beta-catenin, which is enriched at adherens junctions. NMDA receptors also mediate SYNGAP phosphorylation, which then regulates RAS, RAC, cofilin, and actin. It is likely that NMDA receptors and SYNGAP fulfill similar roles at extrasynaptic sites within cerebellar glomeruli. And so this is just a full overview of the results that I found both in the molecular layer and in the granule cell layer, which map allowed me to see the great enough resolution to obtain these results. Thank you.